Recombinant DNA technology is a method used to combine DNA from different sources to create new genetic sequences. This involves inserting foreign DNA into host organism's genome, enabling the organism to express new traits or produce specific proteins. Mendel's groundbreaking experiments on inheritance provided the foundational principles of genetics, leading to the rapid advancements of biotechnology and genetic engineering. Modern biotechnology revolutionized every aspect of human life. Genetically engineered bacteria have been used to increase soil fertility. Genetically engineered crops that are more resilient to diseases and climate change help to address global food shortage. Genetically modified organisms are used for environmental cleanup, such as removing pollutants through bioremediation and creating eco friendly products like biofuels. Biotechnology enables efficient production processes like fermentation and development of biodegradable materials. Gene therapy attempts to repair faulty genes to enable personalized medicine. DNA technology is widely used in criminal investigations and paternity testing. Cloning is a process of creating identical copies of an organism, cell, or gene. Gene cloning is creating copies of specific DNA fragments or genes by inserting them into vectors and growing them into host cells, like bacteria. Cell cloning produces identical cells from a single original cell, often used in research to create cell lines. Organismal cloning produces a whole organism that is genetically identical to its original. This can be done through methods like somatic cell nuclear transfer. Somatic cell nuclear transfer is a method where the nucleus of a somatic cell is transferred into egg cell that has had its nucleus removed. The egg then develops into genetically identical organism. That's how the famous sheep dolly was developed. Through genetic engineering, scientists synthesize recombinant DNA by combining DNA from different sources. It involves the following steps: selecting gene of interest, that is the specific gene to be cloned, molecular scissors or restriction enzymes. Cut the DNA at specific sites to isolate the gene of interest. Molecular carrier or vectors like plasmids carry the gene of interest. The gene is placed onto a vector. The vector containing the genes of interest is introduced into an expression system, such as bacteria, animals, or plant cells. Inside the system, gene replicates or produces a specific product like a protein. This method results in the production of recombinant DNA, leading to the creation of specific products like proteins or enzymes. There are three possible ways to get the genes of interest: to isolate it from chromosomes, to synthesize chemically, and to make it from messenger RNA. Genes can be isolated from the chromosomes by cutting the chromosomes on the flanking or neighboring sites of the gene using specific enzymes known as restriction endonucleases. Another very common method of getting the gene is to synthesize it in the laboratory from messenger RNA using reverse transcriptase. This DNA molecule is called complementary DNA or cDNA. Small genes can be synthesized in the laboratory using chemical methods. This is useful for short genes or segments. Molecular scissors or restriction endonucleases are the natural enzymes of bacteria, which they use for their own protection against viruses. The restriction enzyme cuts down the viral DNA but does no harm to the bacterial chromosome. They are called restriction enzymes because they restrict the growth of viruses. These enzymes cut DNA at exact sequences of nucleotides, called recognition sites. These sites are often palindromic, the same forwards and backwards. When the DNA is cut, it leaves single stranded ends called sticky ends that can easily attach to other DNA pieces. So far, more than 400 such enzymes have been isolated, out of which about 20 are frequently used in recombinant DNA technology. EcoR1 is commonly used restriction enzyme in molecular cloning, DNA mapping, and genetic engineering due to its predictable and reproducible cutting pattern. It cuts double-stranded DNA at specific sequence of bases at the cleavage site. A gap is introduced at which a piece of foreign DNA with the complementary bases can be placed. The single-stranded but complementary ends of the two DNA molecules are called sticky ends. They therefore facilitate the insertion of foreign DNA into vector DNA. Vectors are molecules used to carry a gene of interest into a host cell for cloning or expression. They are crucial in gene cloning, gene therapy, protein production, and transgenic organism creation. Among few common vectors are plasmids, which are small circular DNA molecules found in bacteria, commonly used for gene cloning. Viral vectors are modified viruses used to deliver genes into animal or plant cells. Hybrid vectors are phagemids or cosmids, combining features of plasmids and phages. Artificial chromosomes like bacterial artificial chromosomes and yeast artificial chromosomes are larger vectors used to clone very large DNA fragments. Shuttle vectors, vectors that can replicate in multiple host organisms, such as bacteria and yeast, enabling the study of genes in different systems. To prepare recombinant DNA, the plasmid is cut with the same restriction enzyme that was used to isolate the gene of interest. The gene is joined to the sticky ends of the plasmid. This joining process is facilitated by a special enzyme called DNA ligase, which seals the foreign DNA into plasmid vector. At this stage, the two different DNA pieces are combined, forming recombinant DNA or chimeric DNA. The recombinant genes can be introduced into bacteria through transformation or transduction. Transformation involves making bacterial cell membranes permeable, often using calcium chloride, to allow the uptake of recombinant plasmid. Once inside, the plasmid replicates as bacteria divide, creating clones that carry the recombinant gene and produce the desired product. Transduction, on the other hand, uses bacteriophages as vectors. The phage attaches to host bacterium and injects recombinant DNA into it. The bacterium then produces new phages, each carrying a copy of cloned gene. Screening of vectors often involves the use of antibiotic resistance genes. For example, PBR322 plasmid contains genes for both tetracycline and ampicillin resistance. During cloning, the gene of interest is inserted into tetracycline resistance gene, disrupting its function. After transformation, the bacteria are first grown on ampicillin containing plates, where only cells that have taken up plasmid will survive due to ampicillin resistance gene. These colonies are then transferred to tetracycline containing plates. Bacteria that fail to grow on tetracycline plates indicate successful gene insertion. In this way, colonies that are ampicillin resistant but tetracycline sensitive are selected as positive clones containing the recombinant DNA. A genome is a complete set of genes of an organism. A genomic library is a collection of bacterial or bacteriophage colons, where each colon contains a different DNA fragment from the organism. To create this library, the organism's DNA is cut into pieces and inserted into vectors, like plasmids or viruses, which are then taken up by the host bacteria. The entire library holds all the genes of the organism. To find a specific gene, a probe, that is a single-stranded labeled nucleotide sequence, can hybridize with the target gene. Once the probe binds, the specific gene can be identified, isolated, and further analyzed or cloned. Polymerase chain reaction is a widely used technique in molecular biology that allows scientists to make millions of copies of specific DNA segment. It's a fast and efficient way to amplify DNA for applications such as gene cloning, forensic analysis, and disease diagnosis. This method relies on heat-stable enzyme called DNA polymerase to replicate DNA. The process involves three main steps: denaturation, where the DNA strands are heated to separate them; annealing, where short primers bind to target DNA sequence; and extension, where DNA polymerase adds nucleotides to build new strands of DNA. Taq polymerase is crucial for the success of polymerase chain reaction because it is heat-stable enzyme derived from bacterium Thermus aquaticus, which thrives in hot environments like thermal springs. Unlike regular DNA polymerases, Taq polymerase can withstand the high temperatures, around 95 degrees C, needed during the denaturation step of PCR where DNA strands separate. Its stability allows it to remain active throughout the repeated heating and cooling cycles of PCR. PCR cleverly mimics the natural process of DNA replication but in a controlled, in vitro setting. Like in cellular replication, PCR requires DNA templates, primers, nucleotides, and polymerase to replicate a specific DNA sequence. However, PCR resolves several challenges associated with in vitro replication. It replaces the needs for helicase, which unwinds the DNA in the cells, by using heat to denature the DNA strands. The use of Taq polymerase allows PCR to endure high temperatures required for denaturation. PCR uses synthetic primers that specifically target the desired sequence, ensuring that only the regions of interest are amplified. Polymerase chain reaction has a wide range of applications across various fields. PCR is used to detect pathogens such as viruses and bacteria by amplifying their DNA or RNA. It's used to identify genetic mutations, inherited disorders, and prenatal screenings. It helps in diagnosing conditions like cystic fibrosis or sickle cell anemia. It enables the amplification of tiny DNA samples from crime scene, allowing for accurate identification of individuals through DNA profiling. In molecular biology, PCR is used for gene cloning, sequencing, and studying gene expression, making it fundamental in genetic and evolutionary research. PCR is applied to detect genetically modified organisms, study plant diseases, and improve crop breeding programs. The key reagents used in PCR are DNA template contains the target DNA sequence to be amplified. Primers short DNA sequences that bind to the target region on the template DNA, making the start and end points for amplification. Taq polymerase, a heat stable enzyme that synthesizes new DNA strands by adding nucleotides to the primers. DNTPs or deoxynucleotide triphosphates, the building blocks for DNA synthesis that Taq polymerase uses to extend the DNA strand. Buffer solution. Buffer solution maintains the optimal pH and ionic environment for Taq polymerase activity during the PCR process. The steps of polymerase chain reaction include denaturation. The reaction mixture is heated to around 94 to 95 degrees centigrade, causing the double-stranded DNA to separate into two single strands. Annealing. The temperature is lowered to 50 to 65 degrees centigrade, allowing the primers to bind or anneal to the complementary sequences on the single-stranded DNA. Extension. The temperature is raised to around 72 degrees C by adding nucleotides to the primers, creating copies of the target DNA. These steps are repeated for 25 to 35 cycles, exponentially amplifying the target DNA sequence. Agarose gel electrophoresis is a technique used to separate and analyze DNA based on their size. In this process, agarose gel of the required percentage is prepared and mixed with the ethidium bromide that binds with the DNA to be visualized and poured into casting tray. Gels are created in the gel where the samples will be loaded. DNA samples mixed with the loading dye are pipetted into the wells of the gel. A DNA ladder or marker containing the fragments of the known sizes is also loaded for the reference. An electric current is applied across the gel. The gel is visualized under UV light or imaging systems. This reveals bands representing the different DNA fragments, which can be compared to the DNA ladder to determine the size of the fragment. In agarose gel electrophoresis, DNA moves through the gel based primarily on the size rather than charge. DNA molecules are negatively charged due to their phosphate backbone, so they naturally migrate towards the positive electrode when an electric field is applied. However, within the gel matrix, the rate at which the DNA moves is influenced by its size. Smaller DNA molecules travel faster and further through the porous agarose gel compared to the larger fragments. The gel acts as a sieve where the mesh sizes restrict the movement of larger molecules more than the smaller ones, leading to the separation of DNA fragments by size. DNA loading dye is crucial in gel electrophoresis as it helps in visualizing and tracking the progress of DNA samples during the separation process. One key component of the loading dye is glycerol, which increases the density of the sample, ensuring that it sinks into the wells of the agarose gel rather than diffusing out. This helps maintain the samples in the well and prevent it from dispersing into the running buffer. Loading dyes often contain tracking dyes that migrate at known rates, allowing researchers to monitor the progress of the electrophoresis in real time and ensure that DNA fragments are properly separated and visualized. Types of PCR include several variations designed for specific applications. The conventional form is simple, used to identify DNA segments, following typical denaturation, annealing, and extension steps. Real-time PCR measures DNA amplifications in real time, allowing quantifications of the DNA. Reverse transcriptase PCR converts RNA into complementary DNA or cDNA before amplification. Multiplex PCR amplifies multiple DNA targets in a single reaction by using more than one primer pair. Nested PCR increases specificity by using two sets of primers in two successive reactions. Hot start PCR enhances specificity and reduces non-specific amplifications by using a heat-activated DNA polymerase. Touchdown PCR gradually lowers the annealing temperature to enhance primer binding specificity. On average, humans share 99.9% of their DNA. The remaining 0.1%, about 3 million bases, makes each person unique. These differences can be used to distinguish individuals by comparing short, repetitive DNA sequences that vary greatly among people. Early methods compared sequences up to 100 bases long, called mini satellites, to create unique DNA pattern or DNA fingerprint. Modern techniques now use even shorter sequences called microsatellites. Except for identical twins, it's highly unlikely for two people to have the same DNA fingerprint and is identical across all cells in the body, remaining unchanged by any treatment. Mini satellites are composed of repetitive DNA sequences that are 10 to 60 base pairs long, while microsatellites consist of short DNA repetitive sequences ranging from 1 to 6 base pairs, sometimes up to 10. Mini satellites typically repeat 5 to 50 times within the genome, while microsatellites repeat more frequently, often 5 to 100 times. Mini satellites are primarily found in telomeres and centromeres, while microsatellites mostly located in non-coding regions of the genome but can sometimes be found in coding regions. Mini satellites are rich in cytosine and guanine bases, while microsatellites contain more thymine and adenine bases. Mini satellites are less commonly used in modern DNA profiling techniques, primarily used in early fingerprinting methods. Microsatellites are widely used in modern DNA profiling, forensic analysis, and genetic studies. Short tandem repeats are repeating sequences of 2 to 6 base pairs of DNA that occur throughout the genome. These sequences are highly variable between individuals, making them valuable for genetic profiling. STR analysis involves amplifying these repeat regions using PCR and then separating and quantifying the fragments through gel electrophoresis or capillary electrophoresis. The resulting patterns of STR create a unique genome fingerprint for each individual, which is widely used in forensic sciences, paternity testing, and genetic research. STRs are advantageous because they are abundant in genome, highly polymorphic, and their analysis is relatively straightforward and reliable. Variable number tandem repeats are regions of DNA where a short sequence of nucleotides is repeated multiple times in a row. The number of these repeats varies between individuals, making VNTRs useful for genetic profiling. VNTR analysis involves extracting DNA, amplifying the VNTR regions using PCR, and then separating the amplified fragments through gel electrophoresis to determine the number of repeats. This variability in repeat number provides a unique genetic signature for each individual. VNTRs are commonly used in forensic science, paternity testing, population genetic studies due to their high variability and ability to differentiate between individuals. The Human Genome Project was a large international scientific effort to map and understand all the DNA that makes up human genes. It aimed to identify, map, and sequence all the genes in the human genome, both in terms of their physical location and function. The project officially started in 1990 and was mostly completed by 2003, making it the largest collaborative biological project ever. Planning began after the US government adopted the idea in 1984, and by April 14, 2003, about 92% of the human genome had been mapped. In May 2021, scientists reached the complete genome level, with only 0.3% of the bases having possible issues. And the final gap free version was finished in January 2022. Map and sequence all nucleotides in human haploid reference genome, which contains over 3 billion base pairs. identify and locate all human genes estimated to be around 20,000 to 25,000. The project involved sequencing samples collected from a small number of individuals and assembling these sequence fragments to obtain a complete sequence for each of 23 human chromosome pairs. Establish databases to store and manage these genetic informations for easy access and use by researchers worldwide. Advance new methods and technologies for analyzing and interpreting the vast amount of genetic data generated. Explore and address the potential ethical implications of genetic research, including privacy concerns, genetic discriminations, and informed consent. Emphasizing that much of the human genome is same across all humans, which is vital for understanding human genetics and health. 1984. The Idea for Human Genome Project is initiated in the United States. May 1985. A workshop at University of California, Santa Cruz to discuss creating a reference genome using gene sequencing technologies. 1986. Charles Delacy outlines a plan for the project. 1988. The first line item for the Human Genome Project is included in the President Reagan's budget proposal. 1990. The Human Genome Project officially begins with the Department of Energy and National Institute of Health as primary funding agencies. 2000. A working draft of the genome is announced, showcasing the collaborative effort of international geneticists. April 2003. The project announces the completion of essential complete genome sequence 2 years ahead of schedule. Key findings of the draft of Human Genome Project in 2001 and complete of 2004 genome sequences include: there are approximately 22,300 protein coding genes in human beings, the same range as in other mammals. The human genome has significantly more segmental duplications, nearly identical repeated sections of DNA, than had been previously suspected. At the time when the draft sequence was published, fewer than 7% of protein families appeared to be vertebrate specific. Funding of the Human Genome Project was a significant factor in its success, primarily sourced from US government. Initiated in 1980s, the project received early financial backing from the Department of Energy and National Institute of Health. By 1988, the US Congress approved the first line item for the Human Genome Project in the President Reagan's budget, with a combined commitment from both Department of Energy and NIH. Ultimately, the project was expected to cost around $3 billion, but was completed for approximately $2.7 billion, reflecting effective resource management. Additionally, the funding was supplemented by international contributions such as United Kingdom, France, Australia, and China, which expanded the project's scope and fostered global collaboration in genomic research. There were fears that genetic knowledge could lead to discrimination by employers and health insurance companies. The Health Insurance Portability and Accountability Act was enacted in 1996 to protect against unauthorized release of health information. Established in 1990 to address ethical, legal, and social issues, allocating 5% of annual budget for this purpose. ELSI funding rose from approximately $1.57 billion in 1990 to around $18 billion by 2014. Concerns that mapping the genome could reshape patient expectations and perceptions of illness among doctors. Human Genome Project successfully sequenced approximately 3.1 billion base pairs of the human genome. The first printout of the human genome was showcased as a series of books at the Wellcome Collection in London. Recognized as a mega project, the Human Genome Project began in 1990 with the goal of mapping the entire human genetic information set. The genome was divided into smaller segments, approximately 150,000 base pairs long, which were inserted into bacterial artificial chromosomes. Funding supported large sequencing centers including the Whitehead Institute, Wellcome Sanger Institute, Washington University in St. Louis, and Baylor College of Medicine, facilitating high throughput sequencing efforts. The United Nations Educational, Scientific, and Cultural Organization played a crucial role in promoting the participation of developing countries in Human Genome Project. With the completion of human genome sequence, researchers focused on identifying genetic variants linked to the common diseases like cancer and diabetes. Detailed knowledge of human genome is expected to derive innovations in medicines and biotechnology. Companies started offering genetic tests to assess predisposition to various illnesses, including breast cancer and cystic fibrosis. The genome information is anticipated to enhance the understanding of diseases, potentially leading to better management strategies and treatments. Biologists can utilize the human genome database to gather comprehensive information on specific genes, including their functions, structures, evolutionary relationships, and associated diseases, facilitating targeted research. The project has influenced genomic research in agriculture, leading to healthier and more disease resistant crops.